Hello friends, most welcome in your dream classroom, Hortus Botanica. Myself, your botany mentor, Dr. Mukesh Kumar. Yes, morphology of flowering plants we are studying. Today, our topic is the roots and especially regions of fruit. Yes, very interesting. We have to study what are the regions of a root, right? How and where these different parts of the root are oriented. What is the position of part of the roots? These are the concern of regions of fruit. So let's see about the regions of root. See. Yes. So whenever we will talk about regions of fruit, what happen? We can easily differentiate the regions of fruit into major three regions. Yes, you all know that we have, see, yeah, see. So first of all, we have region of meristematic activity, right, which is being covered by root cap in general, and just above the region of meristematic activity we have region of elongation, and above that we have region of maturation. Yes, so from distal end to the proximal end we can say first Rama. Yes, I used to say Rama for region of meristematic activity. Then Roy, yes, region of elongation, and then Ram, yes, this is the region of maturation, isn't it? Now come, what you are finding that these root hairs are present on regions of maturation, and just at the tip means Rama, region of meristematic activity, you will find that this is a very soft and delicate part of the root tip. That's why it is being covered by a protective covering. Yes, this is the root cap. So let's see first about the root cap. Yeah, see the root cap. So root cap, a cap or thimble-like structure, cap-like structure, protective covering that covers root apex. Okay, so the structure that covers, that protects the root apex is called root cap. This root cap, mind well, it is a multicellular structure. Unlike root hairs you know it's the unicellular structure, but root cap, yeah, it's a multicellular structure. It is mainly made up of parenchymatous tissues. Question came in previous medical examination, entrance examination, that this root cap is made up of, yes, you can say as parenchyma or special tissues that constitute root cap. This is called calyptrogen tissue. It is very common in monocots and supposed to be the fourth histogen, isn't it? Now, what is the function of this root cap? Yeah, it makes its way through soil. How? Through the graviperception, isn't it? So it moves towards gravitation. We studied through the previous lectures that whenever you are finding this is the root tip, so this root tip is covered by this very protective covering, and this very protective covering is called root cap. Now I am telling that this root cap, what happened, will move towards this gravitation. So it has hormones at the root tip that give the perception to move towards gravitation, and this is called graviperception, well described by, yes, Darwin as well as Endicott scientists, by the way. So come, see, whenever you are talking about root tip covering, yes, root tip covering, so it might be possible your plant will be, yes, mesophytes. So if it is mesophytes, then we have root cap, mesophytes. Remember, I told you that we have major three plants: mesophytes, hydrophytes and xerophytes. So see, if you are talking about first mesophytes, then in mesophyte the root tip is being covered by the cap-like structure. See, this is the cap-like structure, so this is nothing rather root cap in mesophytes, right? So it is in mesophytes. But whenever we talk about hydrophytes, second group, hydrophytes, plant found in water, habitat is aquatic condition. So what happened? This protective covering is covered by pocket-like structure. See, this is a condition, root pocket. What happened? You will find that this is very root tip. And you will find that this root tip is being covered by not cap, rather this pocket-like structure, and this very structure is called root pocket or pocket root, right? So is it clear? So this is also called as root pocket, clear, or pocket root, same. By the way, whenever we talk about the plants found in the xerophytic condition, these are called xerophytes. Xeric condition, sand condition is there. No water will be there. Water stress condition will be there in their habitat. Then what happened? Then it is found that, see, these are the multiple root cap. Multiple root cap. Okay, and this is only in the case of xerophytes. Now, correct. What is the need of this thing? See, if we have this root tip in xerophytic condition, then what happened? If you are going to form a single this structure, cap-like structure, then what happened? It may degenerate under high temperature. So nature has given its more protection, 1, 2, or more than 2. So a number of more than 1 or 2, you will find cap-like structure is there. So it has not a single root cap, rather a number of root cap. That's why it's called multiple root cap. And this is very common in, yes, xerophytic condition. Remember, pandanus question came that pandanus, yes, root tip is covered by multiple root cap. Yes, Pistia, Hydra, these are the Hydrilla, these are the aquatic plants. So its root tip will be covered by pocket. If you are talking about, yes, mustard, right, mango, these are the very common and covered by the root cap. So this is a very nutshell description about root tip covering. In mesophytes it is root cap, in xerophytes it's multiple root cap, but whenever we'll talk about the hydrophytes, it's root pocket, isn't it? And once you will find that by any means this root tip covering is being injured, what do you think? Which will regenerate very faster among all these three? What do you think? Is the xerophytes, mesophytes or hydrophytes? What is the main purpose of this root cap? Protection. And the more protected area will be the adverse condition, and the adverse condition will be shown only in the xerophytic condition. That's why the fast regeneration will occur in xerophytic plants. So that's why we have multiple root cap in xerophyte. So their regeneration will be very fastest and very slowest in the, yes, hydrophytes, isn't it? Good. Now come, talk about the different regions. Okay, what we have to study, regions of root As I told you, we have important three different regions. Yes, three different regions. First we have region of meristematic activity. As I told you, I use a term Rama. Region of meristematic activity. See here, region of meristematic activity. By name it is very much clear. Meristematic activity. Meristem means to say to divide. So this is the reason where cell continues its division. Cell division is continuously occurring, isn't it? That's why it's called region of meristematic activity. So the cells is very small. That's why cell is actively dividing, having thin cell wall and dense protoplasm. So these are the cellular characteristics. Their length, it might be 1 to 3 mm, right? And the mode of cell division is repeated mitosis, isn't it? See what happened. We have a cell. This one cell will divide into two, right? And all these cells right now will again divide further. Okay, so this will divide again. This will become matured and this will divide again. It will becomes matured, it will divide again. It will becomes matured, it will divide again. So what you are finding? You are finding, see, correct, one cell, then we have two cells. It is permanent, it will divide meristematic. Again we have three cells, then what happened? These two will be permanent, it will remain meristematic. So you will find in this way that each level number of cell will increase one in each step, right? So mind well, question will come that if you are finding about meristematic cell division, okay, so question will come, number of meristematic cell division will be number of meristematic cells plus 1. Say if it is capital M and if it is small m, so your formula will be as, yes, then our formula will be here, capital M will equal to small m plus 1. Similarly, if you are going to count about the number of meristematic cell, then it will be capital M minus 1, isn't it? Okay, so this is all about the meristematic cell division. So question may come to you. What question may come to you? That say if you have this root tip and if we have formed 100 meristematic cells we have, then find out number of meristematic cell division. Clear? So number of meristematic cell division, capital M. It is say small m. So what happened? Capital M, it will be just reduce 1. So what happened? If we have one cell division, remember 2 cell. This will remain meristematic. This one, okay. So 1, 2, 3 and 4, so on. This will be the number of meristematic cell. Okay, so see, so we have 1 cell, 2 cell, 3 cell, 4 cell. The total number of meristematic cell is 4. And see, total number of meristematic cell division is, yes, 3. So just one number will be less than to its meristematic cell. So total number is 100, so it will be 99 and vice versa. If it is 99, it is 100. Okay, so mind well where it will occurs at root apex. Okay, so this is talk about the root apical meristem or root apex. Okay, or say region of meristematic activity. So the such type of the question may come to you related to the meristematic cell division. So mind well it n. So what we are finding here? We are finding n number of cell division. n number of cell division. We are not talking about the whole generation, rather n number of cell division we are talking about. So in each case 1 is 2, 1 is going to divide into 2. So again its mode is mitosis. So I think you have got all the concept very much clear, right? Now talk about, so this is all about what? Mesmatic of reason of mesmatic cell mesmatic activity. Now very important line, see, Rama, that is region of mesmatic activity, lie distal to the region of elongation. See, this is very important line which is usually confused by students. Right now what happened? Whenever you are talking about the distal end, see, this is the distal end and its proximal end. Okay, proximal end, this is the origin point. So this is the origin point from where it is originating, and this is the free end. Okay, free end, another end, that is the free end, right? Now, so you have to understand that this free end means to say distal end, and the origin point is the proximal end. So what you are finding, we have, yes, we have 1, 2, 3 regions. First one, Rama, lie at the distal end. Okay, this Rome, that is the region of maturation, lie at proximal end. And this very important point, region of elongation, if you will talk, right, from this, means to say from this free end, so it is the distal. So what you find, that this region of meristematic, it is lie at the distal end. So if you are going to compare this ROE, region of elongation, then what will you say? That this region of meristematic activity, yes, this one lie away means to say towards the free end. So this is the distal to the ROE, that is region of elongation. So always mind well, with respect to origin as well as free end, we have proximal end and distal end. Always remember, distal, Rama will find at the distal end, Rome, that is region of maturation, file at the proximal end, and this ROE you can say either with respect to the proximal end or either with respect to the distal end, whatever. So you must have this query concept, otherwise whatever come language of the question you can solve by this crystal clear concept, isn't it? Yeah. Now come to the point, second one, region of elongation. Yes, again this is the region of elongation. So proximal to maturation. So if you are talking, we are taking with reference to this one, so we are going towards proximal end, that's why it is proximal to meristematic activity. Okay, now in this region, in this process of elongation, what happens? There is two very important mechanisms. One is the rapid elongation and another is the cell division occurs. After cell division there will be the elongation. This is rapid elongation of cell. So cell elongation. You can say also that there is a cell elongation or rapid elongation. You can say both same. And vacuolization. Another important step you can add, vacuolization. And this rapid elongation, yes, cell elongation, and during this process, okay, what happened? Extension of cell wall takes place. Extension of cell wall takes place, and it is being facilitated by hormones or genes. Now try to understand what I want to tell you. See, if we have a cell like this cell is going to elongate. Yes, again elongation is there. So this elongation is directly proportionate to the vacuolization. There will be vacuole, central vacuole will be there. Central vacuole are going to enlarge. Okay, by the enlargement of this central vacuole, this is called vacuolization. What happened? the cell wall covering this protoplast, see cell wall, what you are finding? This cell wall is slowly going to extend dead. So this is the extension of cell wall. Okay, so their microfibrils are going to extend it with the help of oxygens. So you are finding here during this process what happens, cells are going to be enlarged. So two process, one as I told you, yes, extension of cell wall, it occurs after vacuolization, isn't it? And both collectively execute, yes, rapid cell elongation or rapid elongation. So what you are finding, if this structure is there, this structure is there, this structure is there, so you are finding how the length is increasing. So this reason is responsible for growth of root in length. So it will increase the length of the root, isn't it? Now the last and very important, this is the region of maturation. It is cell proximal to region of elongation, yes, or you can say it lies at the proximal end, isn't it? Now gradually it differentiates and matures, isn't it? Because it's the region of maturation. Now the matured part has to execute their particular function, so they will differentiate and execute their function on maturation. It is also the site of root hairs. Very important, root hairs. Yes, this root hairs, you know, it is epidermal in origin. That's why it is exogenous. And remember, we studied in the previous class that roots are what? Endogenous. But root hairs, again I am telling, it's epidermal in origin, so it's exogenous. Now, it's thread-like structure, tubular structure, and it absorbs water and minerals from the soil. So this is the major and main part, root hairs, that is going to absorb water and minerals from the soil and supply to the root to shoot. So these are the important regions of the root. Okay, so see, at the rate of NCRT, the regions of root, what it is being illustrated, right? Three regions, yes, maturation, elongation, and rama. Yes, we studied about the root cap, protective covering of root tip. So yes, thimble-like structure, root cap, we studied. Few millimeter, we studied how this root has mesomeric activity. Very small cells are there, thin wall, dense cytoplasm, we studied. They execute mitotic cell division. Cells proximal to this region undergo rapid elongation and enlargement and responsible for length in root, right? We already studied. So after that we have elongation. Okay, that elongated cell will differentiate and matures and form, yes, maturation that lie at the proximal end. Okay, and we know that we have root hairs that is epidermal in origin. They have very fine and delicate thread-like structures that absorb water and minerals from the soil. So this is all about the region of roots at the rate of NCRT. So I think we have got very much good, clear concept about the region of root, their proximal and the distal origin, as well as their function. Thank you very much. Keep watching, like, subscribe, and give your good comments to make you more available such types of the true and original videos. Thank you very much.